All right, good afternoon. Welcome to Consilium. My name is Lisa Spienko. I head Consilium Scientific. And today we're hosting our second event with Cancer Research UK. We're bringing together a fascinating panel of experts to discuss the challenges of screening for cancer and specifically on the use of surrogate endpoints. Today's event will be chaired by Professor Bethany Shinkins from University of Warwick. She's the member of the UK Screening Committee, and she's also a senior advisor for NICE. So Bethany, at this stage, I'm handing the panel into your capable hands, so please go ahead.
Thanks very much, Lisa. So thanks for that introduction, and thank you to Cancer Research UK and Consilium for inviting me to chair this webinar. So today we're going to be discussing the use of surrogate endpoints in cancer screening trials. So we've got a fantastic lineup of speakers who I know have all individually spent a lot of time thinking about this topic. So I'm going to start by asking each of them to introduce themselves. And when you do that, can you also declare any relevant conflicts of interest you may have? So Hilary, do you want to go first?
Sure, I can go first. Hi, my name is Hilary Robbins. I have no conflicts of interest to declare. I'm an epidemiologist. I'm originally from the US and I'm trained in cancer epidemiology and biostatistics. I currently work at the International Agency for Research on Cancer, which is located in Lyon, France, and we're part of the World Health Organization. My research is focused mainly on cancer screening and early detection. So I work in a few specific contexts, including lung cancer screening and biomarkers for early detection of lung cancer. How can we use biomarkers alongside lung cancer screening? Also, biomarkers for early detection of HPV-driven cancers. And then finally, the specific topic we're here to talk about today, sort of understanding evidence from cancer screening trials and thinking about standards of evidence for novel tests. At IARC, I co-lead a team called the Risk Assessment and Early Detection Team, which is, I think we're about 15 people from 12 different countries at the very moment.
Thanks, Hilary. Peter, do you want to go next?
Hi, I'm Peter Sassini. I'm a professor of cancer epidemiology at Queen Mary University of London. The conflicts are that I have run a number of cancer screening trials, most of which have not used cancer mortality as an outcome. So several studies in HPV testing or self-sampling and cervical screening, which certainly haven't used cancer mortality. I'm running the BEST 4 trial of a sponge device for collecting cells from the esophagus in esophageal cancer screening, which has an outcome which includes reduction in stage 2 or worse cancer. And perhaps the biggest conflict is NHS Gallery, because that is sponsored by a commercial company called Grail, and that trial too uses advanced stage cancer as the primary outcome.
Okay, thanks, Peter. Sian?
Hi, I'm Sian Taylor-Phillips. I'm Professor of Population Health at Warwick University, and I do a range of evidence synthesis and primary research in screening. I have no conflicts to declare, no money from industry to self or my institution. I'm funded by an NIHR research professorship primarily, which looks at evidence synthesis methods for screening policy, and surrogate outcomes is a part of that. I am a UK National Screening Committee member, and I chair their research and methodology group. And part of the remit of that group is to think about what sorts of evidence will be most useful for the UK National Screening Committee. But anything I say today is very much my own views and not those of the UK National Screening Committee or NIHR. Thanks.
Okay, thank you. So very, very comprehensive. So, okay, thanks everyone.
Sian and I both have a grant from Cancer Research UK, which Sian leads, for looking at surrogate outcomes within cancer screening trials.
Absolutely, yeah. And I've been involved in that too. And again, for me, nothing I say is representative of the UK NSC. Okay, I think we've got all that covered. So thanks. I think you'll all agree we've got a great panel today, lots of expertise in this field, so we should be able to have a good discussion. So I'm going to start by providing a brief introduction to the topic so that we're all on the same page, we're all starting in the same place. And then each panelist is going to, in turn, kind of put forward their position on the topic. And then we've got some specific questions for our panelists. And throughout the webinar, you're going to be able to post your comments and questions in the chat. And I will be also asking for people to raise their hands and ask their questions themselves. And that'll be towards the second half of the webinar. Okay. So let's start very broad. What do we mean by cancer screening? So cancer screening involves systematically testing apparently healthy people who may have a higher risk of cancer with the goal of detecting cancer at an early, more treatable stage, or to identify precancerous conditions before they progress to cancer. So this proactive approach aims to intercept cancer early in its progression, increasing the chances of successful treatment and better outcomes. So in the UK at the moment, we've got currently 4 national cancer screening programmes in bowel, cervical, breast, and a new targeted lung screening programme, which is currently being rolled out across the UK. So the goal of cancer screening is to reduce cancer-specific mortality and morbidity. Now, to determine whether a screening programme is effective, researchers rely on randomised controlled trials. So these trials typically evaluate long-term outcomes such as reductions in cancer-specific mortality or overall mortality. But the problem is these endpoints can take years or even decades to materialise, making the evaluation process lengthy and resource intensive. One of the issues is that by the time these studies have been completed, the test technology used to originally screen people has often been improved upon, or something else might have come along which is likely to be better. It's also possible that cancer treatments have improved over that time, meaning that some of the results of the trial may not be representative of what could be achieved in current real-world clinical practice. So this is where surrogate endpoints come in. So for some cancers, they could be potentially helpful. A surrogate endpoint is a measure that stands in for a clinical outcome of interest. And in the context of cancer screening, it should predict the effect of screening on cancer mortality. So data for surrogate endpoints tends to be quicker to obtain than mortality data, and therefore provides earlier signals of whether a screening intervention is effective. So this can accelerate trial timelines, reduce research costs and resources, and facilitate faster decision-making about the implementation of new screening programmes. So the use of surrogate endpoints in cancer screening is a hot topic because their adoption would fundamentally change the goalposts for screening programmes. This shift is contentious because there's debate about whether there are any endpoints which reliably predict cancer-specific mortality. There's discussion around the level of evidence required to demonstrate this and how generalisable this evidence is when thinking about different cancers and different screening contexts. So in summary, surrogate endpoints offer a promising pathway to streamline cancer screening trials, but there is debate about whether they should be used, and if so, which ones and when. Okay, so now I'm going to invite each of our panel members to provide a brief overview of their position on this topic, talking about what evidence has been produced to date for surrogate outcomes in the context of cancer screening and what's their interpretation of this evidence. Hilary, do you want to start?
Yeah, sure, I can start. So, thinking about the evidence that's been generated so far, I mean, in my mind, there's sort of 3 categories in a way. The first is sort of conceptual development around this entire idea of how surrogate endpoints can be applied to cancer screening, which it's sort of evidence in a way. I think the conceptual development is quite important. The second category perhaps is modeling evidence. So sort of simulation modeling of what happens during cancer screening, and trying to use that to understand what different potential endpoints might do in the setting of a trial. And then the third category, where I've been involved specifically, is analyzing data from prior trials. So looking at prior screening trials and how different potential endpoints, what we've observed about different endpoints in those trials, and trying to bring that information together to learn something that we can apply in the future. So, in conceptual development, Peter's been a leader there, I would say. You know, we need to first sort of enumerate the different potential surrogate endpoints that we're even talking about. And, you know, the one that we most commonly focus on is the idea of reducing the rate of late stage cancer. And this is very importantly different from the idea of sort of reducing the proportion of cases that are detected at early stage. You know, the proportion of cases detected at early or late stage is importantly influenced by overdiagnosis or lead time bias. Whereas if we reduce the rate at which people are diagnosed with late stage cancer, then that's a much more robust endpoint. There's other endpoints that are proposed. I won't go through them. And then, you know, in the area of modeling, you know, researchers in this area have aimed to sort of predict the cancer mortality reductions observed in prior trials based on the observations about the reduction in late stage cancer. And then, so if we say, okay, we reduced stage 3 and 4 cancer by X amount, we use information about stage specific survival. So how long do people survive if they're diagnosed at one stage or the other? What mortality reduction would we expect to have observed in that trial? And what mortality reduction did we actually observe? And that area of evidence has delivered sort of mixed findings, I would say. Sometimes we see what we expected and sometimes we don't. And then my team has what we've done primarily is a sort of systematic review looking at prior cancer screening trials. And it's very simple, really. What we did is we looked at in each trial, by how much was the rate of stage 3 and 4 cancer reduced, and by how much was the rate of late stage cancer reduced, and then looking by each cancer type, breast, colorectal, lung, prostate, and ovarian. What is the relationship between those two endpoints? And what we observed is that the tightness of the relationship is different depending on which cancer type you're looking at, and also the slope. So the amount of the reduction in late stage cancer that seems to translate into reduced cancer mortality, that seems to vary across cancer type as well. So when you take it all together, I think what's very clear is that the way that potential surrogate endpoints relate to the definitive endpoint, which is cancer mortality, is different for different cancer types. And that makes it very complicated if I mean, we're not always talking about multi-cancer early detection tests, but if we are, then it's very complicated to think about surrogate endpoints because we're combining together different cancer types in which endpoints behave differently. So that's my take for now.
Okay, thanks, Hilary. Peter.
Yeah, so as Hilary says, I like to think of it conceptually. So I think first we need to formulate the problem a little bit more clearly. Are we looking for a surrogate for testing or a surrogate for estimation? For estimation, the usual trial endpoint, targeted cancer mortality, is merely a surrogate for quality adjusted life years, which is typically estimated through health economic modelling and is what most guidelines or groups make their ultimate recommendations based on. Hence, I think it's reasonable to say that we're interested in surrogates that can be used instead of mortality for testing purposes. And that's important because that's why we wouldn't expect the same relationship. You know, some of what Hilary was saying doesn't work. There's no reason why it should work because we're not trying to talk about estimation. And so, you know, we can still use that surrogate endpoint as part of modelling to then predict whether it's mortality or quality adjusted life years. One potential surrogate, which was proposed in the context of the UK flexible sigmoidoscopy trial, was predicted mortality. So the idea is that if we could reliably estimate cancer mortality up to a given date based on the features of the cancers at the time of diagnosis, that would firstly yield an answer much sooner and secondly be less subject to random noise and therefore yield a more precise answer because you're saying on average how will the people do rather than saying, oh well, in this small sample I happen to have this number of people who died or didn't die by a certain date. And I'm not really aware of systematic empirical study of that endpoint, but I think people are looking at it and they're definitely thinking about it. Essential in predicting mortality at the time of diagnosis is the stage at diagnosis. For all types of cancer, the more advanced a cancer is at diagnosis in terms of stage, the more likely it is to be fatal. Cancer screening works by detecting a cancer earlier. As Bethany said, the aim is to intercept earlier. So the point is that with that earlier stage, it should be more likely to be successfully treated. So thus there's a logic model behind using advanced stage as a surrogate for mortality. And if screening works, it will almost certainly result in a reduction in advanced stage cancer. Further, there's an advantage that by separating the stage from the post-diagnostic survival, one can separate the effects of screening, which is the early diagnosis, from the effects of treatment, which means that if you want to know how will screening that was studied 10 years ago be performing today, given that treatments have changed, you don't necessarily need to redo all those trials, because actually the trial is the combination of the screening and the treatments that are around then. So when might advanced stage fail as a surrogate? And I think there are two key things. One is when there is no or very little survival difference between early and late stage cancers, or when only a small proportion of cancer deaths originate from late stage cancers. And the second is when, stage for stage, screen detected cancers have worse prognosis than non-screen detected cancers. And in particular, when, regardless of whether screen detected or not, stage-specific survival is worse in screened individuals than never screened individuals, and potentially when you've got cancers which are so-called born to be bad, doesn't matter when you find them, they're going to be fatal. So in practice, this issue about a little difference between advanced and early stage cancers, or very few advanced cancers, explains why stage 4 cancer is a poor surrogate for ovarian cancer, for instance, and why historically advanced stage was a poor surrogate for liver cancer, because in those trials everyone with liver cancer died within a few years. And why, unless you have very long follow-up, advanced stage may appear to be a poor surrogate in prostate cancer, because so few of the cancer deaths will be from advanced cancer, and so few people, even with advanced cancers, die within the follow-up that many trials have. So what of the evidence? So plotting the relative reduction in mortality against the relative reduction in advanced stage, which is what Hilary did, and she found 41 trials in her meta-analysis. There are very few outliers, and I would argue that all those outliers can be excluded because of reason one, that there is you don't anticipate that it will be a good surrogate because of what you already know about the survival. Plotting the Z value for mortality against the Z value for advanced stage, which is, rather than plotting the relative risk, the Z value is telling you how much information you have to say that screening works. And if you do that from meta-analysis of each type of screening, so you have 1 point on your plot for each type of screening, that gives you a very, very strong correlation. You get a plot that, except for the liver cancer example, virtually everything seems to fall on a straight line. So there's 12 types of screening with RCT evidence. 5 are non-significant on both outcomes, so the Z is less than 2, and 6 are significant on both outcomes. And as I said, the only exception is liver cancer, for which you would say that, well, there is just the survival from early stage liver cancer is so poor that that's not going to work. So I think the evidence to say that if what you're trying to do is to say it's a surrogate for testing, does this type of screening work, yes or no, then the evidence that so long as you think about when you're using it, it's a pretty good surrogate.
Okay, thank you, Peter. And finally, Sian.
What a fun spot to follow Hilary and then Peter. So I'm going to try and focus on the bits where we agree. So I think we all agree that including any sort of early stage cancer is problematic because of overdiagnosis, right? And you can't have early stage cancer in your numerator, obviously, but also not in your denominator, right? Because you can't look at the proportion of cancers that are late stage. I think we can easily agree that. I think we all agree that this relationship between late stage cancer and mortality is dependent on cancer type. And of course, it's dependent on cancer type because survival by stage differs by cancer type. So necessarily it must be different. So if you're trying to develop a sufficient surrogate for a different type of cancer, and that's what we really want to do, we want to do it for a different type of cancer, then that's when it's really useful. Then late stage cancer on its own can't be enough. You need to somehow account for these differences in stage, either, as kind of Peter says, by just intelligently ruling out things like liver cancer, where you haven't got a difference in survival by stage, or at least not in those trials. Or you need to look at something like predicted mortality, where you're factoring in these things into your metric. But then when you do that, you have to just be careful you don't accidentally bring early stage cancer back into your metric. The other complications that I want to add into the mix is it's not just cancer type, it's test type. So most of these trials that we've talked about are imaging tests. And so they are trying to detect the cancer when it is in the original organ. How does that change when we're looking at blood-based tests, multi-cancer early detection tests? How does that relate? I don't know the answer, but we definitely need to pose the question. And then also differentiating between screening programs that work by stopping cancer, by preventing cancer, and those that whose mechanism of action is detecting cancer early, and whether that has any implications for our surrogate. And then I just wanted to reflect on, well, how do we know if it's sufficient? And I can sort of understand Peter's point of view. You look at these correlations, there really is a correlation, right, between trials. It tends to be that those trials where they reduce late stage cancer, they tend to reduce mortality more, right? That there's an unquestionable correlation. It's just you don't want to be making policy based on an outlier to that, and can we explain all of the cancers that don't fit within that? And we've only got 50 odd screening trials, and so we have to be reasonably sure that we've understood all of these mechanisms before we make a leap based on it. And as a measure, correlation is very hard to interpret. And when you look at sort of treatment trials, cancer treatment trials, where most of the literature around surrogate outcomes is, they have metrics that I find intuitively a little easier to understand. And Peter probably knows by now that I'm really interested in a surrogate threshold effect, right? Which is the minimum treatment effect on the surrogate that's reliably predictive of a treatment effect on mortality. And if you think about that, if you're thinking from a kind of policymaker perspective, if you have something like that, you can kind of interpret it. Whereas if you have a correlation of 0.7, how do you decide if that's enough? That's all I have to add.
Okay, so you've left us on a really good point there. So how are we going to actually produce this evidence efficiently? So we know, I think all of us have all worked on analyses of historic screening trials. Now, the majority of those historic screening trials are on cancers where we've already got a screening program. So actually, when we're starting to think about cancers where there isn't a current screening program, how are we going to actually produce this evidence of sufficiency or adequacy, whatever word you want to use, of a surrogate outcome in those contexts? So we know that there's differences between cancers, And we've talked a little bit about the fact that that relates to the survival differences between cancers, but also the context and the type of tests. So thinking prospectively, okay, so we've got a new test in a cancer that we haven't got a screening program in, and there may not be much historic evidence in terms of past screening trials where we can look at correlations. What are we going to do in those contexts?
So I would argue that by the time we get the sort of evidence that Sian would like, we don't need a surrogate outcome anymore, because she's quite right that it depends not only on the site of the cancer, but also on the mechanism of the test, what it's looking for. And so it's true that many of the new blood-based tests, the fact that they're shedding cell-free DNA is probably a hallmark of a more aggressive cancer. So unless we say we will never accept a surrogate endpoint, you're never going to get the evidence that you want, because by the time you've got it, you've done 2 trials and you've shown what the answer is, so you don't need it. So I think the issue is, how do you incorporate what you believe to be a pretty good surrogate into the evidence generation pathway in order to accelerate that pathway without totally relying on it, if it happens to be that possibly for the first time ever, combining advanced stage with predicted mortality is giving you the wrong answer. And so you want to be able to see that you could then change the course of development. But if you don't use a surrogate, you end up doing some trials for 15 years. All the resources are put into those trials when I think it was pretty clear earlier on that they were not going to be successful, and you could have sort of switched resource. And secondly, the development time for cancer screening has been ridiculously slow. So the main trials taking 9 to 15 years to publish on mortality, faster for more rapidly growing cancers and longer for the slower ones. And then typically another 10 to 15 years before you've got rollout of a screening program. So, is it 11 years since NLST published, Hilary? And 16% uptake of lung screening in the USA.
It was 2011. We're getting old, all of
14 years. So even after you get the mortality outcome, we're still messing around before we actually start to do something, and that is not good enough. You're not going to get any companies to invest in new biomarker testing if it's going to take them 20 years from starting a trial to being able to get to market. And so, unless you accept the only screening we've got, the screening we've got now is all we're ever going to do, you need to be saying we have to be a bit more pragmatic here, and what on the sum of evidence looks like the most promising thing to be going forward with.
So I don't think it's this kind of binary. You're either terribly cautious and you never implement anything, or you just accept that late stage is great and you go for it. There's sort of grades in between, isn't there? I think so. Several things you brought up, Peter. The gap between the end of the trial and the implementation is something that can be addressed, because, for example, you can use surrogates not just for a yes/no decision, but you can use surrogates for sort of deciding about piloting and pathways and all that sort of thing. It's there's 2 levels of decision, isn't there? There's a decision to kind of pilot and get yourself set up versus a decision to implement something. And the sort of the worry about the decision to implement something is, once you've implemented cancer screening, it's almost impossible to de-implement it. So it depends on the cancer screening, but often if you have, for example, screening that has a lot of overdiagnosis, then you have the popularity paradox, where a lot of people believe it saved their life, but actually it overdiagnosed them. So it is challenging, and you want to be quite sure before you roll out a large new screening program. But there's also a nuance within multi-cancer early detection tests, because yes, BowelScope was discontinued, but bowel screening wasn't discontinued. And I'm not suggesting it should have been, because it's a really good screening program. But it is difficult to roll back screening programs. So then within MSEDS, there is another approach where you have one big trial that looks at mortality over a group of cancers. So what's the cancer-specific mortality for this group of cancers that you tested for? And then you use a series of surrogates to look at what each of those cancers within that group has contributed. And that helps you understand whether actually within that test you've got the right group of cancers.
I wanted to clarify sort of at the beginning of the answers to this question that what we mean is a surrogate endpoint that's sufficient for implementation, right? And clearly, Sean, that's how we took the question. And, you know, I tend to agree that I know many people don't agree with this idea, but in my view, cancer screening is special. It's very different from cancer treatment because it's something that we do across the entire population of healthy people, not a specific, very small group of individuals who are already sick. So I think that, my view is that we do cancer screening, the individuals partake in cancer screening because they believe that it might save their life, or it might at least extend their life. And based on what we've learned so far about surrogate endpoints, I remain unconvinced that we're going to find one that's going to be good enough. There's a lot of really creative ideas coming out about, testing samples from the control arm and looking at test positivity among people who died of cancer across the two arms, this kind of thing. And which is, this is exactly the kind of thinking that I think we need. But still, it doesn't seem to be a game changer. It doesn't allow us to take a trial with a cancer mortality endpoint and make it really tiny, or make it really inexpensive, or make it really fast. It doesn't, none of the ideas that have surfaced seem to achieve any of those things. And, more and more, I'm starting to think that maybe instead of a surrogate endpoint, what we need is a way to be really, really, really sure that a test is going to work before we do the trial in the first place. And that the trial isn't going to fail, because cancer screening trials fail to show cancer mortality benefit all the time. And the type of work that my team does on early detection biomarkers leverages large resources of banked samples, so banked pre-diagnostic samples. I do start to wonder if a way forward is to really understand exactly how a test needs to perform. If we're talking about blood tests, or even imaging-based tests could use this strategy as well, using banked images. Really understand exactly how a test needs to perform for different stages at different lead times for different cancer types and banked samples, so that we can, quantify that performance and be really sure when we launch a trial with a cancer mortality outcome that it's going to be successful. And I don't agree that we need 20 years for a readout of a cancer mortality trial, especially for a multi-cancer early detection test that works. If we have one that works, it saves lives, it reduces cancer mortality. You have the benefit of aggregating multiple cancer types together in that cancer mortality outcome. And in theory, we're talking about rapidly fatal cancers like pancreatic cancer, for example. Lung cancer is a very important player, and the potential benefit offered by multi-cancer tests, again, rapidly fatal cancer. We don't need 20 years. We should be able to deliver that trial evidence in 6-7 years, but perhaps we just need to be really, really sure that that test is going to work before we start. Okay, Peter's raising his hand.
Just
Okay,
how would you
can I just
design a
pause
trial
there?
for prostate screening? Okay, sorry.
Sorry, can I just pause there? Just to say to the audience, if you've got questions that you want to ask in the next, we've got 25 minutes left. So if you've got questions that you want to ask in person, if you can put your hand up, and if you can, or write them in the chat, it's up to you. But we'll come back to your questions. But if you can start thinking about what questions you want to ask, that would be great. Sorry, Peter.
So I was just wondering how Hilary would apply that to prostate cancer screening. It sounds to me that you're having a surrogate that's even less closely linked to mortality. If you're using biobanks to study biomarkers, because you're not even intervening at all, you're just seeing what would have happened had you done the test earlier. So the direct, how it relates directly to mortality is one step removed. Of course, it's a great idea to do as much as that before a trial as possible, but if you don't have a surrogate for mortality for prostate screening, I can't see how you could do a trial in under 15 years.
If you don't have a surrogate to do a trial for prostate screening,
because you need to wait, the deaths tend to be many years after diagnosis.
Indeed, if we're talking about less aggressive, less rapidly fatal cancers, it takes a while for those trials to read out. I do agree with you.
Which
but I think for
causes more death
multi-cancer
than any other type of cancers.
Yeah. But for multi-cancer tests, we're talking about a mixture. We're talking about a mixture of cancer types, some of which are rapidly fatal. And just to be clear, what I'm suggesting is not at all that retrospective evidence, retrospectively obtained evidence from prospectively collected samples would be sufficient to implement a test in the population. What I'm suggesting is that, maybe we need to better understand these tests and then do a really definitive trial, because the evidence we have about surrogate endpoints so far isn't suggesting to me that we can make these trials as small, fast, and cheap as we would really want them to be in order to be doing many, many, many of them, for example.
Okay, Sean?
Yeah, to me, it's a really difficult problem, right? Because everyone would like to implement effective screening earlier, right? I think that's sort of universal, and it's kind of balancing those risks of delaying effective screening with those risks of implementing ineffective screening. And I want to know more about Peter's view, particularly for MSEDs, when it is it's a different type of test. At the end of a trial, and you're the trialist here, if you've got a reduction in late stage cancer from MSEDs, would you want to see implementation based on that? Is that your position?
So if you had a statistically significant reduction, which I think gets to your threshold effect, because you're only going to have that if it would meet a threshold, I would then want to see a pilot implementation, which would be happening to see what is needed to do a new type of screening program. What happens in practice? How do we cope with doing that much screening, the workup? It could totally change the demands for types of treatment. That lung screening means that we need more surgeons than we used to for treating lung cancer, because we're finding more early lung cancers. Yeah, that takes a lot of planning to train up the extra surgeons. So you can be doing that fairly large scale piloting of the implementation whilst you're waiting for the mortality data from the trial and collecting more data. One of the data that's key is to see whether or not a cancer that is screen detectable, how that affects the survival. So if you find, in breast screening, once you adjust for stage, there's very little difference between survival between screen detected and either interval or never screened cancers. except yeah, it's slightly better if you're screen detected. The worry is that with a lot of these new blood-based tests, the survival will be worse if you're screen detectable than if you're not. So you need to be able to make that adjustment. But you can put it all together as the predicted mortality. So you'd have your randomized controlled evidence to say what impact is it having on stage, and then you can have cohort studies and biobank studies which will tell you how it will affect survival, and you get a better stage and screening test result prediction of survival, and you can put that together to infer what mortality would be. And so needing to be using results like that, I mean, the NHS gallery is 140,000 people with 3 rounds of screening, and we recruited in a year, and there'll be 3 years of follow-up, so 4 years to do the trial. The proposal from the NCI is 200,000 people, 5 rounds of screening, and then a minimum of 5 years of follow-up, because it will take 2 years to recruit. So it's a 7-year trial. Instead of a 4-year trial, it's 200,000 instead of 140,000, and because per person you've got more tests, so the cost of the trial will probably be 3 times what NHS Gallery has costed. I don't know what it cost, but NLST was about $250 million. So these trials are huge. If you could get the results 2 years earlier, that's big. If you could do it for half the cost, that means you could do 2 trials instead of 1. And as I say, for all 12 types of screening that there's been randomized controlled trials, using advanced stage would have given you the right answer based on individual levels, except for liver, and I would argue you should never have done it with liver. So,
Peter, can I ask as a follow-up question as well? In the scenario that you described with sort of large scale piloting, um, you know, the outcomes in the original RCT continue to accrue, and there's and eventually you look at cancer mortality, and there's different potential scenarios. One is that you see a statistically significant reduction in cancer mortality. Beautiful. One is that you see in a sort of modest reduction in cancer mortality. It's not statistically significant. It's somewhere sort of, in the middle. But then there's a scenario in which there's clearly no cancer mortality benefit at all, which could be caused by, for example, differences, like you said, in the survival of screen detectable and undetectable cases. So, what is what action would you suggest in each of those potential universes, where, depending on what we see with the cancer mortality result?
So in the 2 extremes, I think it's easy. If you see a significant reduction in mortality, I don't think you have to worry about multiple testing. Go ahead and make sure you do the health economics and everything else, and try and roll out that screening program as quickly as possible. If you see no reduction in cancer mortality, and there's a clear inconsistency between the stage shift results and the mortality results, you've got to pause. You do not continue with your pilot implementation. You need to understand what was going on. Why was it? And the chances are you would never introduce a full-scale screening program unless there's some oddity that you've understood and you realize that's why. The middle case is the hardest, and it's a question of what evidence do you want. And I think because it's what I've said is that essentially the NHS gallery will be underpowered for mortality, so you would quite expect that fairly often you would have shown a significant result for advanced stage, but you wouldn't get a significant result for mortality. So I think you'd be looking to see, is it consistent rather than is it significant. I've thought that maybe you would use a one-sided test. So because you're really looking for confirmation. So, do you need to still have the same type 1 error that you had? You could be sort of saying, well, it's a provisional thing, and we want to continue to see these things. But I think some of it would be you also want to collect this survival data to see what's going on. But it's a little bit like these early marketing that drugs get before they've been shown definitively. But you need some way of being able to start collecting the data. And then, if you could do your pilot implementation through a cluster randomized, the sort of things that they do in Nordic countries where they introduce it into different areas at different times, and so that you could get a fairly rigorous evaluation from those about what the impact on cancer mortality would be. So you can sort of say that we will, within a certain amount of time, have the evidence that we really want. Going back to your earlier point, of course I agree with you that screening is quite different because we're approaching a seemingly healthy population rather than waiting for people with symptoms to come to us to say, help me, I've got a problem. Nevertheless, I do think that sometimes we are overly concerned about the harms of screening. And if you haven't seen harms of screening, to say, oh, well, you still need really, really high quality evidence, much higher than we'd be requiring for something else for treatment, where we do know there's terrible harms very often. I think it's a little bit out of kilter because it's the same sort of things that would happen with vaccination. You cannot do a trial and find something which is going to affect 1 in 100,000 people. And yet that may happen with a vaccine. You may have a terrible, severe side effect reaction to a vaccine that's very rare. So you won't get that from the randomized controlled trials. So similarly, to think about everything that could possibly go wrong, essentially you're doing a test. If that test is not dangerous, these theoretical problems you will see if you start doing a huge amount of excess surgery that you didn't need to do. You'll get that from the trial. And so you should then be able to say, actually, I know that the harms aren't major. There's a cost and I need to think about that. But to say, oh, it's absolutely forbidden to introduce screening, to offer screening to people if you don't know for certain that it's going to have a mortality benefit. And I think there's also an ethical thing. For me, if I saw that you'd reduced all advanced stage cancers by 12%, I wouldn't be equipoise anymore. I would not be willing to randomize someone and say, I'm going to get a blood sample from you, but I may not test it. Because, I think, and the other thing is if we are curing people of advanced stage cancers, so there's no mortality advantage, I would still prefer to be diagnosed and cured of a stage 1 cancer than to be diagnosed and cured of a stage 4 cancer or a stage 3 cancer, because simple surgery I would prefer to having some new biological drug and eventually being cured.
So does that apply to you? Would you apply that to liver cancer as well then, where there isn't a survival advantage?
So they're saying this is if you're going to be cured in any case. If you're going to die in any case, I'm not sure there's any advantage of having it earlier. I mean, it's something that you'd want to find out from patients. They sometimes do prefer to have more time to plan things, and a very rapid death within 6 weeks of diagnosis is hard. But you probably go through all the same nasty treatments, nasty death, whether you were first diagnosed at stage 1, but it just keeps progressing. But I'm saying if you're going to be cured, and with all these new treatments, more and more people are living 5 years and more after diagnosis of advanced cancer. That's where I'm saying I would have preferred not to have had to have those really aggressive treatments.
Okay, Thanks, Peter. So I'm going to pause there and ask, so we've got 10 minutes left. I haven't seen any hands or questions come through on the chat. So we've got 10 minutes, which probably gives you about 3 minutes each to really focus your message down. And I want you to focus on thinking from speaking to researchers and then separately speaking to policymakers. So what decisions or actions are needed next? And that might be in the short term or in the long term. Okay, so if you were thinking, putting your research hat on, what do you think is needed next, if anything? And if you were thinking about speaking to policymakers or you are a policymaker, what actions do you think are needed? Hilary, do you want to go first? Slightly put you on the spot there.
I'll try. I think so. If I tried to speak specifically to the question, which is about surrogate endpoints, because if we broaden and we start talking about cancer screening evidence in general, it becomes a very big discussion. I think if we really want to understand surrogate endpoints, for sure, we need more research. We are not there. Like I said, in my personal view is that we are not there. We have not found a surrogate endpoint that we can simply pop in for cancer mortality and move on. But it is, in thinking about it, it's a bit challenging to say exactly what research is going to be definitive. In fact, I think there's more that we can learn by bringing together data from trials that have been done in the past. I don't think we've exhausted that avenue. I think there's still more to learn. But, I come back to my earlier point that maybe a better way forward is to quantify exactly what a biomarker, what a test needs to do in banked samples. And there we have a lot of learning to do to sort of link performance characteristics to screening benefit for different cancer types. It's a very complex problem, but if we really, really understood that, then maybe we could make excellent decisions about which tests should be moved forward into large-scale randomized clinical trials with the cancer mortality endpoint. So in thinking about it, that's where I sort of land. I suppose at the end, I'm a bit pessimistic about surrogate endpoints, I guess I have to say. Speaking to policymakers, this topic is really complex. And I think that the biggest problem we have is that detecting cancer earlier sounds like a good thing. And it is not always a good thing in the sense that it does not always extend life. And that is a difficult concept. So I suppose the first thing I would say is that essentially detecting cancer earlier doesn't always extend life. Here's why. We've discussed some sort of frameworks for how to move forward, how to think about, okay, trials and then pilot implementation and then trial evidence. And I think more kind of dialogue on this is needed so that we can have more of a consensus about what policymakers expect cancer screening to deliver and what the public expects cancer screening to deliver and how much uncertainty we're willing to accommodate and accept as we move from one decision point to the next, trials, implementation, and so on.
Okay, thank you. Can I just double check? Charles Bennett apparently had your hand up.
Yes.
Have you still got a question or not? Yep, we can hear you.
Can you hear me?
Yeah.
Yep. Okay. Thank you. So you asked about whether you want to talk from the research end or the policy end hat. So where I'm asking for, so I can declare my background, I am a medical oncologist who does research and clinical work in prostate cancer. I have a PhD in public policy, so unfortunately, I'm going to cross both. And the third perspective, which you didn't bring, is I unfortunately also have prostate cancer. So I'm all going to talk about the perspectives, those tri perspectives. And so for me, particularly in prostate cancer, and I'm just going to prostate cancer, when I think about prostate cancer and screening and late stage prostate cancer, a late stage prostate cancer that's a high Gleason score is much different to me than a late stage prostate cancer that's a low Gleason score, because a low Gleason score will be very responsive to various treatments we have up there, and we have a long-term survival, and you'll never die of your cancer. So this whole discussion has talked about dichotomous of late stage and non-late stage cancer, and I would like to subset late stage cancers into high-grade prostate and non-high-grade prostate, because I want to screen out high-grade prostate cancers.
Okay, thank you very much. Peter, Sian, have you got anything to say in response to that?
That's what we did in our review, because we share your view.
I mean, I would always agree that you'd like more. I think the reason we've done our trials the way we have, I mean, it wasn't prostate, is to have a simple endpoint. And we thought that if we had some complex combination of different stages, a weighted thing of different stages, and taking into account grade or the molecular phenotype, and doing things differently for different cancers, it would just be too open to say, like, it's not very transparent. You know, it's, if you've got deaths from cancer, it's very easy. You can say how many there were in each arm. If we were just talking about advanced cancers, it was simple. But you do want to take that into account, and you do need to look at whether the advanced cancers that are there have a much worse prognosis because they are all high grade or something. So I would agree.
Absolutely. Thank you for that question. So you've got 1.5 minutes each. Peter, do you want to go next?
So from a policy perspective, I think policymakers need to learn what they learned in COVID. They need to make decisions based on imperfect evidence, and waiting for the perfect evidence is a route to stopping progress in cancer screening, because we'll never get that evidence. And then to go with that, they need to think much more carefully about how they can introduce pilots so that they can then be withdrawn without ever introducing a full screening program, so that you don't get these problems. So thinking about how should we do this so that society benefits, rather than thinking this is a one-off decision we're only going to make once, we need to wait till we've got the perfect evidence. For researchers, I think there's a lot of things that we want to try to understand the relationship between the screening test and the biology, to understand whether the sorts of cancers that we're detecting are quite different. I actually think that, whereas I agree with Hilary that there is not an advantage of detecting cancers earlier for many cancers, I don't think it's true that preventing advanced stage cancer is, it's almost always a good thing. And the only time it's not is when even the early stage cancers are really bad prognosis. But if we're going to improve treatments for those early stage cancers, that's mostly in cancer types where we never see cancers early. And the only way we're going to start developing new treatments is if we start finding those cancers earlier. And so that's not an argument to have a whole screening program, but we do need those biomarkers and get those tests being used more in order to find more early cancers to improve treatments. And if we do find that the biology is different, then you'll probably get targeted treatments. And so already people are thinking with cell-free DNA, should you escalate treatment if you have cell-free DNA with an early stage cancer, or de-escalate it if you've got an advanced cancer with not?
Okay, thank you very much. And very quickly, Sian, and then we are wrapping up for this evening.
Very quickly, what researchers need to do, I think we urgently need to invest in the research methods to look at this. You know, you've got decades of this research in surrogates in cancer treatment, and people haven't invested to translate that to the more complex intervention of cancer screening. And we're spending tens of millions of pounds on trials, and actually we should be investing in the methods research. And then for policymakers, I actually think researchers, as a researcher, we need to do a better job of supporting policymakers with this, because what we're essentially doing at the moment is showing them correlation coefficients. And I think that's giving them a pretty impossible task.
Okay, thank you very much, and thanks for being quick. Okay, so we are out of time. I want to thank all of our panelists for taking time out of their already long working day, so appreciate that, and thanks for making my job as a chair extremely easy. You handled all the debate between yourselves, which was fantastic. So I think we've all learned a lot. I certainly have, and I hope you've all enjoyed it. Thank you for joining us. Bye.
Thanks, everyone. Bye.